So let's say we have our cell, and we know inside of our cell we have our nucleus with our genome. And we know our genome has genes. For example, maybe we have this blue gene, which creates a very specific protein, and maybe we have this red gene, which creates another protein, and maybe we have this purple gene, which creates another protein. But we know the way this process works. For example, we have this blue gene, which has a very specific nucleic acid sequence, which gets transcribed into a very specific mRNA, which then gets translated into a very specific protein. Maybe this gene creates some kind of antioxidant protein. Then we have this red gene, this very specific nucleic acid sequence, which gets transcribed into a very specific mRNA, which gets translated into another very specific protein. Maybe this gene creates a protein that deals with low oxygen concentrations. And again, we have another gene, this purple gene, which again creates a very specific mRNA, which now maybe creates a protein that manages low glucose concentrations. But the point is, these genes encode proteins. These genes create proteins. However, it's important to realize this process of taking our DNA, transcribing it into mRNA, then translating that into a protein, this process requires a lot of ATP. This is a very energetic process, which requires a lot of energy. So therefore, we know we have a lot of genes in our genome. So at all times, should we be expressing all of our genes in our genome? Well, no, that's a waste of energy. For example, what if we have high oxygen concentrations? If we have high oxygen concentrations, should we go waste all the energy of taking this gene and creating these proteins, which manage low oxygen concentrations? No, that's a waste of energy. So therefore, it's very important to regulate which genes are being expressed to create these proteins. So how do we do this? Well, it differs for all genes, but a very common theme you'll see in cellular biology is this. So let's say we have this gene which we know creates some kind of protein that deals with oxidative stress. So should we express this gene at all times? No, we should only express this gene if we have oxidative stress. So how do we regulate that? Well, first, let's say we have this sensor, this dimer, this heterodimer made out of these two different proteins. So we have this sensor that's sensing oxidative damage and oxidative stress. So now let's say for whatever reason, we have some oxidative stress. Maybe we've ingested a poison, or maybe our mitochondria are malfunctioning, whatever. For whatever reason, in our cell now we have oxidative stress. Now we have this oxidative stress, so now we should create this protein to deal with that antioxidant protein. So how does that happen? Well, this oxidative stress causes a conformational change in the sensor, which causes it to dissociate. Now it dissociates into these two individual parts. But this part happens to be a transcription factor. And these transcription factors are very important. It's these transcription factors which regulate whether or not a gene is activated. Once this transcription factor gets activated, now it can initiate the transcription and expression of this protein. But before, remember, when in the normal conditions without that oxidative stress, this transcription factor was sequestered. It was binded to this purple part, so it wasn't active. However, we went through oxidative stress, which dissociated the sensor, now dissociated. Now the transcription factor is free. Now that it's free, now it can enter inside the nucleus and initiate transcription. That's why it's called a transcription factor. So now it initiates transcription of this gene, which we know creates an mRNA. Now that mRNA gets processed, now it enters into the cytoplasm and now creates some kind of antioxidant protein. So now we've created this antioxidant protein which can deal with that oxidative stress. And now we're only expressing this gene when we need it. We're not just constitutively always expressing this protein. We're only expressing it when we need it. And that's a way to save energy. And it's very important to regulate these genes. We don't want to just express these genes under all conditions because we know that that's a waste of energy. And it's that common theme. We know we deal with oxidizing conditions. It activates this transcription factor, which now initiates the process of expressing this gene. Maybe we have low oxygen concentrations. So under low oxygen concentrations, maybe they cause some kind of conformational change, which now activates this transcription factor, this red transcription factor, which can now enter the nucleus and initiate the transcription of this gene, and now we're starting to express this gene. Or maybe we have low glucose concentrations, which somehow activate this transcription factor, which now enters into the nucleus and now stimulates and initiates the expression of this particular gene, which handles that low glucose concentrations. So these transcription factors are very important. They regulate which genes are active or not. If this transcription factor is not active, then we're not going to create and express this gene. And this is a common theme you'll see in biology. So now, here's something really important. So let's say we have our endothelial cell with our nucleus in our genome, and let's say we have our hepatocyte with our nucleus in our genome, and let's say we have a neuron with our nucleus in our genome. It's important to realize every single cell in our body, for example, in our body, every single cell in our body has the exact same genome. We know we get chromosomes from our mom and our dad, but every single cell in our body has the exact same genome, whether it's an endothelial cell or a hepatocyte or a neuron. The only exception is certain immune cells and also in our gametes. But besides that, nearly every single cell in our body has the exact same genome. So if they all have the exact same genome with the same genes, why are they so different? Why is this endothelial cell very different from this hepatocyte? And why is this hepatocyte vastly different from this neuron? We can tell by their morphology and their functions. We know they're very different cells. But why are they such different cells if they all have the exact same genome with the exact same genes? Well, it's not which genes they have, it's which genes they're expressing, which determines what kind of cell you have. So for example, maybe this endothelial cell has a lot of these activated red transcription factors. So now this endothelial cell has a lot of these red transcription factors that are activated. So they're going to essentially initiate the transcription of this gene. And so this gene is going to create a lot of these proteins. So this gene is very active and expressed in this particular cell. However, in a neuron, it has none of these activated red transcription factors. So therefore, there's no transcription factors to activate this gene. So therefore, this gene is not expressed, and therefore it's creating no proteins. And maybe this hepatocyte is somewhere in the middle. Maybe it has some activated red transcription factors. So it's going to activate and initiate some expression of this gene, creating some protein. But maybe we have a different transcription factor. Maybe this purple transcription factor is highly activated in hepatocytes. So therefore, they're going to highly express this gene, creating a lot of this particular protein. And maybe this neuron has very little of this activated transcription factor, so it's activating very little of this gene, creating very little of this protein. But we see all cells have different combinations of activated transcription factors, so they're all going to have different combination of which genes are expressed and which proteins they have. And it's that difference in those combinations in which genes are expressed which determines what kind of cell you have. So that's why we have such different cells. And maybe you probably understand the theme now, but maybe this neuron has a lot of activated pink transcription factor. So therefore, this gene is very highly activated, and it's creating a lot of this particular protein. And so it's these different combinations of proteins and activated transcription factors which determine what kind of final cell you have. But the point is, it's these transcription factors which regulate which genes are activated. So even though all cells have the exact same genes, that's not what's important. What's important is which genes are actually being activated and transcribed to create very specific proteins. And the combination of proteins determines what kind of gene you have. But there are certain proteins that all cells need. For example, all cells go through glycolysis. So therefore, the proteins needed for glycolysis are needed for all cells. For example, maybe this gene creates the proteins for glycolysis. So therefore, in all of these cells, they're going to activate these proteins. And there are certain proteins and certain genes we like to call housekeeping genes, which are needed just to keep any generic cell activated. So all of these cells are going to be activating those proteins. And something else important to realize are these transcription factors are not static, they're dynamic, they change. Which transcription factors are activated and which genes are expressed changes. For example, maybe this hepatocyte releases some kind of protein. Maybe it releases some kind of protein, or maybe a glycoprotein or some kind of molecule. So this molecule enters the bloodstream, now it hits our endothelial cell. So now it hits our endothelial cell and it binds to this receptor. So now it binds to this receptor, and once it binds to this receptor, it stimulates some kind of signal transduction cascade, where now it binds to this receptor goes through a conformational change, which activates the chemical reaction. which then activates another chemical reaction, which activates another chemical reaction, which eventually activates this yellow transcription factor. Now this yellow transcription factor is activated, so now it can turn on this gene. So now this gene is now activated, now it's going to start creating this protein. So originally this gene wasn't activated and wasn't creating protein. However, this molecules triggered this cascade, so now this gene is activated. So which genes are expressed is dynamic. It changes with time.